No, I literally
Okay, before we start today, just a quick advertisement for Dr. Cleary's medical microparasitic mycology course this fall. He thinks he has enough students to run it, but he would like to have some more. So if you can at least consider signing up for that, that would be great. We've only talked about one mycoses in this class, so a lot more interesting out there. Hopefully you all, not only, I'm probably preaching to the choir here, but hopefully most of you watched the lecture that I posted from last week and saw that extensive announcement that I put up yesterday. So we're gonna, hopefully, if I get to be going too fast, just kind of wave your hands around and stop and ask some questions by all means. But we're gonna try to finish the innate immunity today. Maybe in start adaptive, we'll see where we're at. I'll do what we can on Monday and then post a lecture hopefully Wednesday nights, and then we'll use Monday more of a kind of catch up day and questions, and we'll probably do some new material that day that won't be on next Wednesday's exam. So I know that's come up quick. I think it will work out. It will be a little shorter exam, still worth 100 points. So I think that'll work out good. On that lecture that I posted, probably the most complicated thing is complements. I know it's a lot of like letters and numbers and what does this mean? Remember, complement proteins, they're just proteins that are circulating in our blood. And when they become activated, they can go out into the tissues where the site of infection may be. They're cascade reactions. So one protein acts on another, which acts on another. And they have several different functions. One, they will increase inflammation. We're going to talk more about inflammation today. Some of those complement bits like C3b act as opsonins. What are opsonins? Yeah, what do you think?
Don't they help with like phagocytosis?
Right, they help with phagocytosis. Right, good. They make phagocytosis like more likely to happen, right? So C3b complement fragments can be opsonins, antibody can be opsonins. We'll talk about those. And some of those complement fragments can come together to form membrane attack complexes. So we'll do some review questions again with complement on Monday. If there's no other questions before we get started, let's move on and talk about the kind of cells that usually are the first thing that you might think about when you think about our innate immune response. And those are the cells that we have circulating in our bloodstream, and those leave the bloodstream as well. Your book calls them formed elements in the blood. Okay, so think about these kind of cells, think about where they came from. All these formed elements start out with some stem cell in the bone marrow. Okay, and then by a process of differentiation we call hematopoiesis. Okay, again, that's just means by a process of differentiation. Those stem cells can develop into different kinds of stem cells, myeloid stem cells and lymphoid stem cells. The myeloid stem cells will differentiate into the platelets, which we mention them here just because they're there in the bloodstream, but they really don't have much of an immune function at all. They're important for clotting blood, of course. And red blood cells, erythrocytes, they don't have an immune function either. Of course, their job is to carry oxygen throughout our body. And one important thing about erythrocytes or red blood cells, they have a nucleus when they're formed, then they lose that nucleus somehow. And I don't know enough about it as like how that happens or why that happens. But that's sort of an interesting scenario later on in terms of thinking about how we can understand if that erythrocyte has been infected with some kind of pathogen. The normal way that it advertises whether it's infected is not there for those type of cells. So we'll come back to that. We're not going to really talk anymore about mast cells, but those are present. Those are considered a white blood cell as well. And then these myeloblasts will differentiate into a group of cells that we call the granulocytes. Whoops, not done down there. It's a little bit better. Sites. The basophils, neutrophils, and eosinophils, those are considered granulocytes because they have granules, and we'll talk about a little bit what's inside those granules. And then monocytes, which when monocytes leave the bloodstream, they will differentiate into either macrophages or dendritic cells. Okay, so the monocytes, along with the lymphocytes here, these are considered agranulocytes. A meaning without, okay? So they don't have those granules. And those T lymphocytes and B lymphocytes, again, they differentiated from a different kind of stem cell there, along with natural killer cells. And I'm not really sure what to call natural killer cells. In the book here, they talk about how they have granules, but we don't really consider them granulocytes. So I wouldn't really worry about that they have granules. We're gonna just mention those really, really briefly, not pay too much attention to them. Whether they're granulocytes or agranulocytes, those are all considered white blood cells. And another name for those are leukocytes. So it's kind of inherently confusing when you have many different names for the same thing, but we'll try to do our best to keep track of that. All right, so let's look at these different kinds of cells in a little bit more detail and talk about what they do. I do show you pictures of them, but I wouldn't ask that on an exam. You know, I'll show you a picture and ask you what kind of cell that is. That's if you take a histology class, then you'll learn how to do that. We can differentiate them by using different dyes, though, but we're not going to worry about that. The first kind of granulocyte that we think about are the neutrophils because they're the most common white blood cell in the bloodstream. Okay, 50 to 70% of the total white blood cells are going to be neutrophils. So again, that's in the bloodstream. So they really dominate. They're dominant kind of cell in the bloodstream. They are also known as PMNs, and that stands for polymorphonuclear leukocytes. It's kind of a mouthful. So polymorphonuclear leukocytes. That name makes sense if you look at the cell. It's got these many-lobed nuclei. Okay, so polymorphonuclear many-lobed nuclei. And actually, the basophils and the eosinophils have that same kind of nucleus, but because neutrophils so outnumber the basophils and the eosinophils, usually when we're talking about a PMN, we just mean neutrophils. Okay, and it depends exactly, you know, the context of that, but usually that term is used synonymously with neutrophils. These cells are highly motile. Okay, so when they leave the bloodstream, they're going to move by amoeboid action out into the tissues, wherever they're going to follow some chemical signal to wherever the pathogen is. And they are short-lived. So maybe even less than 24 hours before they're replaced. Okay, and that's probably not a problem, usually not a problem, and we'll talk about why that is and why that might be best, that they're actually short-lived. Okay. So what do neutrophils actually do? They kill invaders by 3 different ways. By phagocytosis, and we're going to look at that whole process. We've talked about phagocytosis before, but we're going to look at that process in a little bit more detail. Basically, they're cells that can eat, okay, so they can get rid of invaders that way. They have granules. We said they're granulocytes, so they can degranulate onto pathogens. What kind of things are in those granules? The same sorts of things that we'll find in lysosomes of other white blood cells. So digestive enzymes, some inflammatory mediators as well. So we'll talk about that a little bit more. But this is the reason why it's best that neutrophils are short-lived. Best that they're short-lived. Because when they degranulate and get rid of those digestive enzymes, that's out in our tissues, and that's going to be harming our tissues and our host cells, just like it would some kind of pathogen. So we want that cell maybe to degranulate, but not to just keep degranulating extensively, because again, that's going to cause a certain amount of tissue damage. It's collateral damage as well. And the third way that neutrophils act on pathogens is fairly recently discovered, maybe 10, 15 years ago, the formation of nets, and that stands for neutrophil extracellular traps. Okay, so they found that neutrophils have a way to actually sacrifice themselves in order to immobilize pathogens. So they can actually digest their DNA, their own DNA, their self-DNA. And if you've ever done some kind of mini prep in a lab, DNA can be quite sticky. Okay, so they actually digest their DNA and then kind of explode themselves basically onto pathogens. So you can see this kind of webbing around. This is the Klebsiella pneumoniae in a mouse lung. You can see this netting, almost like, you know, Spider-Man immobilized it or something. But that neutrophil, again, is digested its DNA and kind of hand grenaded itself onto a pathogen. And there's also some antimicrobial compounds associated with that netting, which is basically DNA. Yeah, question. Yes, so that netting is actually the DNA of the neutrophil. That's, you know, and if it can immobilize the pathogen, then it can't, you know, invade deeper into the host. So immobilize it, and then other aspects of our immunity can maybe finish the job. This picture of phagocytosis, I didn't post this. This is, if you Google phagocytosis, this is Wikipedia's picture of phagocytosis. It's supposed to be a neutrophil that's phagocytizing an anthrax bacterium. So it's kind of a cool picture, but the neutrophil should be like way, 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 way bigger than the bacteria. So I'm sort of puzzled by that, but okay, kind of a cool picture anyway. And then this is just another graphic that just shows the 3 different ways that neutrophils are acting: by phagocytosis, by degranulation, and here they call it netosis. Okay, neutrophil extracellular traps. There's some proteases that we didn't really talk about, and then that are associated with that expunged DNA. All right, the other 2 kinds of granulocytes, eosinophils and basophils, I thought we'd talk about them on the same page because their function is very much overlapping. They're a very small percentage of the total white blood cells in circulation, less than 1% for basophils and maybe 1 to 3% for eosinophils. Primarily, both of those kinds of cells are going to act just by degranulation. It appears that the eosinophils can potentially be phagocytic at times. We don't think that's their primary function. Okay, they don't do any kind of that neutrophil extracellular traps. And both are going to play this big role in targeting eukaryotic pathogens. Okay, so a eukaryotic pathogen might be some kind of helminth worm, like a tapeworm or a roundworm, something that's too big to be phagocytized for sure. So if we have a patient that's dealing with some kind of infection and we find that we do a count of their total, what kind of cells they have in their blood, and they find that their eosinophils are elevated, that gives us a hint that either that patient is struggling with some kind of eukaryotic pathogen or they're having an allergic reaction. That's the other possibility. So there's also this big role in inflammation and allergies that we don't well understand how to, dampen that down. That's going to be an inappropriate response. Okay, so similar functions, again, not as common as the neutrophils. This picture I wanted to show you. I don't think I posted this. I don't have this cited very good. This picture actually showed up on my Pinterest feed, and I still laugh about that. Like, they have their algorithms so fine-tuned. Like, I get crochet and recipes and bacteria and stuff like that on my Pinterest. So that's really weird. But I haven't been able to figure out where this picture was originally posted. But this is showing the immature stage of a blood fluke. And it's surrounded by a bunch of eosinophils. All right, so maybe just one eosinophil degranulating wouldn't make much of a difference on some, multicellular pathogen, but you get a bunch of them, all these digestive enzymes, and they're gonna do their job. So kind of a cool picture. All right, and then a little overview of the agranulocytes. We're going to talk about the lymphocytes more next time as we get into adaptive immunity. But the lymphocytes are pretty common in terms of total white blood cells, but most of them, like their name suggests, are not in the bloodstream. Mostly they're circulating in the lymph. So we'll talk more about the lymphatic system later. Maybe like 2 to 6% something of the white blood cells circulating in the blood. So not a lot. Again, just as kind of an overview, what do B cells do? They are going to differentiate into what we call plasma cells. And it's those plasma cells that are going to secrete antibodies. Okay, so B cells, when they get activated, and we'll talk all about how they get activated, they mature into what's called plasma cells, and it's the plasma cells that are pumping out lots and lots of antibodies. And we say that the B cells are important in humoral immunity. And that might be a term that's maybe not used as much anymore, but our body's humors are our body's fluids. So mostly we're thinking bloodstream. B cells are secreting antibodies that are in the blood and our mucous membranes. T cells, there are several different kinds of T cells that we'll talk about more later. T helper cells and cytotoxic T cells. They are going to function in what's called cell-mediated immunity. Basically, those cells, those cytotoxic T cells, are acting on host cells that are infected with some intracellular pathogen. So like if a virus is inside one of our cells, it's the cytotoxic T cell's job to destroy that host cell so that the virus can be destroyed. Okay, so more specifically about that later. I want to just kind of mention briefly natural killer cells. When we first identified this type of cell, we didn't understand how they acted on pathogens. They didn't act in the same way. They didn't recognize invaders with the same mechanism that B and T cells do. So we just called them natural killers. They just naturally do it. We don't understand how. And now we understand a little bit more that if cells are not, all of our cells are regularly displaying, we have a mechanism that we'll talk about to, it's called MHC proteins, to regularly display what's going on inside of the cell. And it's kind of a way for patrolling cells to see that, oh yeah, everything's okay in the cell. They're just advertising what's going on. If that cell is not displaying what's going on inside it, that's when the natural killer cells will act on it. Okay, so some viruses can downregulate our host cell's ability to advertise that everything's okay, and that's a mechanism they have to hide out. Okay, so we have these natural killer cells that go by a little different signals there in order to figure out that there's a pathogen in that cell, or maybe it's a cancerous cell that's dividing uncontrollably. This is the type of cell that's going to help with that. Okay, so they can function in both innate and adaptive immunity depending on the circumstance. Okay. So this is getting a little bit ahead of our discussion, because in your book here, they go ahead and talk about how natural killer cells will recognize cells that are infected. We're going to talk more about this, but here's a healthy cell of the host that's just advertising its antigen by these MHC1. And you can see that the NK cell will ignore this healthy cell. Here's a cell that's not advertising anything, and so the NK cell recognizes a little different activation molecule and will act on it to kill that infected cell. And it acts a similar mechanism that cytotoxic T cells act. Some of the same cytokines are released. All right, so. All right, and then the other kind of agranulocyte, we talked about the B cells and the T cells in particular. We said the monocytes will differentiate into both macrophages and dendritic cells. Okay, so monocytes, again, pretty small percentage of total white blood cells. These cells are fairly long-lived. When the monocytes, again, migrate into the tissues, they differentiate into these macrophages and dendritic cells. Macrophages are kind of, I would say, the most famous phagocytic cell. That's their main job. When they phagocytize an antigen, they are going to process and present antigen. Now, I know I didn't define antigen yet. We're going to get to that. That's the parts of pathogens that our immune system recognizes. They're going to chew that up and present part of it on the outside. So here's, here's a macrophage. That's kind of a shorthand symbol for a macrophage. And it's phagocytizing some, bacterial cell there, we'll say. So here's the macrophage. It's got that bacterial cell inside a membrane, and we'll talk about the steps of phagocytosis, but it's going to, the lysosomes are going to fuse there and hopefully kill that pathogen. Well, then that macrophage has a way to kind of display on the outside, again, by these, it's actually MHC2 receptors. And that macrophage is then going to travel through the lymphatic system and try to make contact with some B or T cell that will now form an adaptive response to whatever pathogen that macrophage was dealing with. So we'll get more into the details of how that happens, but when we talk about processing and presenting antigen, okay, macrophages are really good with that, as well as our dendritic cells. We consider macrophages to be kind of the body's garbage collectors. So they're moving around through the whole body and chewing up dead and dying tissues and cells in our body. Like the neutrophils, if they only last 24 hours and then they die, okay, something's got to come along and kind of clean all that up. Okay. And macrophages are present in many different kinds of tissues. Sometimes a macrophage that's present in a certain part of the body, we even have a special name for them. Like the macrophages in our brain are called microglial cells. Again, lots of macrophages in lots of parts of the body. We have alveolar macrophages, ones in our lungs. They are going to communicate with cytokine signals. Okay, we're not going to worry too much about which cytokines that they secrete. There's probably several different ones. And macrophages are pretty long-lived when we compare them to neutrophils, at least. They are typically live for months. Okay, so again, the neutrophils that are degranulating, we don't necessarily want them to keep degranulating. Macrophages, no granules, so that's fine. They can just keep doing their thing for a long period of time. Okay, so dendritic cells, again, much overlapping function. They're phagocytic. They also process and present antigen. If you look at a dendritic cell, it looks very different. Okay, it's going to use these long branches. You kind of think of branches of a tree almost to present antigen. That's going to travel through the lymph and present antigen to B cells and T cells. They also, dendritic cells are a little bit more common. They're going to patrol the skin and the mucous membranes. Okay, so very common there. Okay, and here is, you know, I'm always on the lookout for good animations. If you ever find some, send them my way. But that's kind of a cute little animation of kind of talking about macrophages and dendritic cells and the difference between them and what they do. All right, and then the figure in your book that kind of just runs through all those different kinds of formed elements in the blood, their major job. It leaves some of them out, like mast cells, like we're not going to worry about that. But again, just kind of their major function. You know, don't worry so much about the numbers present. We talked about kind of relative abundance. And then just to kind of start getting an idea of what they're actually doing, or, you know, where they're moving, right? So we've got these cells in our bloodstream, especially during an inflammatory reaction, which we're going to talk about inflammation soon. So here, if say we had a cut in our skin and some maybe bacteria got into that cut, there's these inflammatory mediators. That's what these little dots are here, are being released, perhaps by the pathogen itself, but also by the tissues around where those pathogens are, calling other cells that are kind of whizzing by in the bloodstream to get out and, you know, leave the bloodstream and get out into where that infection is taking place. So C5a, that's again a chemotactic factor. It's calling those neutrophils and those monocytes out of the bloodstream to come and work where the infection is, and then they're acting by degranulation or nets or phagocytosis, you know, however they work best. Okay. All right, so before we get to inflammation though, I want to talk a little bit more carefully through the steps of phagocytosis. Get a little bit more detail than what we've talked about before. And I can't remember if I changed the order of this. I know the next chapter, I kind of switched my order around quite a bit from what the book is. I think I kind of kept pretty much with it for innate immunity. Okay, so a cell like a macrophage or a dendritic cell, if it's going to eat a pathogen, it has to be where the pathogen is, right? So it has to chemotax, it has to move towards that pathogen. Again, it's going to follow chemical signals from other cells that are releasing cytokines and things to get it there. Okay, so it's going to move, if it's out of the bloodstream, it's going to move by amoeboid action. And then this is a step that we kind of forget about, that macrophage, for instance, has to be able to recognize that that pathogen is not self. It has to recognize that it's something that's worthy of attack. Okay, so there are receptors on the outside of many different kinds of cells. Like macrophages, I think B cells have them as well, dendritic cells. They have pathogen recognition receptors. So your book calls those PRRs. And a specific kind of pathogen recognition receptor are called toll-like receptors. And that's kind of a not intuitive name. The name comes from the people that discovered these receptors said, hey, these remind us of toll receptors that are in Drosophila, that are in fruit flies. And toll is a German word for something that I forget. So it's not intuitive, but what toll-like receptors are, they are, as I mentioned, they're proteins that are in the membrane of phagocytic cells. So especially things like neutrophils, macrophages, and dendritic cells. Again, I think they're more widespread from that, but especially in those kinds of cells. And what these toll-like receptors do, there's about 10 different kinds, we think, in humans. So TLR1, TLR2, so on and so forth. They recognize these broad-based patterns that that pathogens will have that our cells, our host cells, don't exhibit. So things like peptidoglycan or flagellin or lipopolysaccharide, those are molecules that bacteria have that we would never have. Okay, so these broad-based patterns, they call PAMPs. Okay, I know the abbreviations get to be a little suffocating sometimes. That stands for pathogen-associated molecular patterns. Again, so for example, things like peptidoglycan or flagellin or lipopolysaccharide, just to name a few. Okay. And when those toll-like receptors recognize PAMPs, then that's going to not only tell the cell that, yes, this is something that should be phagocytized, we should get rid of it, but it's kind of like a burglar alarm. Like it will send out signals to lots of other cells, like, hey, there's a pathogen here, there's an invader. We need to, start some inflammation. We need to maybe get a complement activated, lots of other things to, lots of other mechanisms of immunity to fight this invader. Okay, so this is your book's figure. Not crazy about this figure, but anyway, this is the one that I posted. So here's in green here, these are the toll-like receptors. So TLR, I'm not sure what they mean by lipopeptide, but TLR4, for example, will recognize lipopolysaccharide. That's sort of the most famous one. And people that figured that one out, they got a Nobel Prize for it. So Again, we think there's about 10 toll-like receptors in humans. They've discovered 12 in mice, so there may be a few we haven't found yet. But again, broad-based patterns that we find on pathogens like Flagellin or peptidoglycan that you wouldn't find on our cells. Some of the toll-like receptors, here's one that's expressed inside the phagosome. Okay, so remember, once the pathogen is phagocytized, there's some that get expressed on the inside of that membrane. And then as the pathogen gets kind of chewed up, there are some toll-like receptors that recognize things like double-stranded RNA or single-stranded DNA, things that would never be found in the host cell. Okay. And so those, once the toll-like receptors recognize those PAMPs, it sends signals to the nucleus to start, again, calling in other aspects of immunity like interferon production, and send cytokine signals to other cells to kind of act on these pathogens. Okay, so this figure, again, we're talking about the steps of phagocytosis. We talked about the chemotaxis, the phagocytes say this is a macrophage, has to move where there's an invader. It has to be able to, this figure kind of skips out the whole recognition part. So say that, yeah, the toll-like receptors have recognized that this is an invader. It has to kind of really grab a hold of that invader. It has to stick to it. And we talked about sometimes that's really hard. Maybe the invader has a big capsule around it and makes it hard to get a hold of. If there's some opsonin present, that adherence of the macrophage to the invader is going to be a whole lot easier. So again, an opsonin, any factor that enhances phagocytosis, it could be complement fragment C3b that acts as an opsonin. It could be an antibody. And we'll see how the pseudopods will have kind of receptors to be able to grab a hold of antibodies a whole lot easier. Okay, so if those pseudopods can stick to the pathogen, more likely that it's going to get ingested. So number 3 there, ingestion of the microbe. It's going to be ingested in a phagosome. Okay, so that reminds me of one of the questions on the exam. I forgot to kind of mention the exam, but I did post a short PowerPoints of some of those exam questions that were most commonly missed. One of the exam questions asked something about viral entry, and it talked about if the virus is enclosed in a membrane, what kind of entry is that? And a number of you said that's membrane fusion. Okay, if the viral membrane fused with the cell membrane, that would be membrane fusion. But here, phagocytosis is endocytosis, where it's enclosing that pathogen in a membrane. Okay, and then when that's a viral entry, that membrane quickly falls away along with maybe the membrane of the virus and the capsid of the virus. But during phagocytosis, so the pathogen is very important that it's enclosed in a membrane, because the lysosomes, which lysosomes are just bags of enzymes that were produced in the rough endoplasmic reticulum and then enclosed in a membrane by the Golgi. Okay, so then those lysosomes are going to fuse with the phagosome and make that, we call that step a phagolysosome. Okay, and these we don't want to get those lysosomal enzymes. We don't want to get them out in the cell, because those would cause problems for that cell itself. And we'll talk about some of the different, well, let's talk about that right now. We'll come back to this. What kind of enzymes are in a lysosome? Well, there's probably about 40 different enzymes, and we'll just talk about a few of them. Lots of digestive enzymes. So enzymes that digest proteins, proteases, or they digest lipids, lipases. Lysozyme, remember, that's an enzyme that breaks up the peptidoglycan, those bonds between the sugars of peptidoglycan. Nucleases, digesting nucleic acid. So lots of others. A couple of other interesting ones. There are respiratory burst enzymes enclosed in that lysosome. So remember we talked about reactive oxygen species. We can actually make reactive oxygen species in our phagosomes of our phagocytic cells. So things like superoxide ion, hydrogen peroxide, hydroxyl radicals, those are oxidizing agents. They're going to grab electrons off of other molecules. So that causes damage to pathogens. Neutrophils have this really interesting enzyme called myeloperoxidase, which actually takes hydrogen peroxide produced up here by one of these enzymes and chlorine and makes hypochlorites. What's another name for hypochlorite? I heard some of you say it, bleach. Okay, so we can actually make bleach in our neutrophils. Okay, so the so what is that most of the time, maybe 99% of the time, that pathogen is killed in that phagolysosome. Okay, those lysosomal enzymes react and usually bring about the death of the pathogen. And then it's going to be eliminated by exocytosis. Now, most of those lysosomal enzymes are actually designed to only be functional at low pH. Okay, so we've got some proton pumps that make that phagosome have a low pH. So when those leftover lysosomal enzymes are gotten rid of, you would think it might do harm, out here to other cells, but because the pH is more neutral outside of the cell, then they don't work. So it's a pretty good system. Okay, and lastly, I just mentioned that actually most phagocytes, especially macrophages, have sort of two levels of activation. They kind of have their normal level where they're just going around our body patrolling and eating stuff. And then when they encounter a pathogen, they become activated, okay? Through cytokine signals, they become activated and they get bigger and they have a higher metabolism and they are just acting a lot more quickly. So this is kind of a cool time-lapse photography here of activated macrophages. And if I can get,
here, what you're looking at is a video of macrophages that have been given yeast to play with. And what you can see is the macrophages will move towards the yeast and then engulf them. So here one is moving directly towards it and then engulfs it. Here's another yeast that's here, which will eventually be attacked and eaten by another macrophage, shown right there. So in much the same way that Metchnikoff found these invading microorganisms activated the macrophage, turned it on, increased its capacity for migration, its capacity for cytotoxic killing, and as shown here, its capacity for actually killing and eating the invading microorganisms. So another move showing that
macrophage is moving around, so it's kind of cool. All right, questions about that? Okay, we're moving fast. Okay, so finally, I've talked a lot about inflammation, hoping that you know a little bit about it already, but let's talk more about why inflammation is so important. Most of us think about inflammation as being a bad thing. And if you see, here's a person that has a condition called chilblains, where basically they have an inflammatory response to cold weather. So in the winter, this person's toes get swelled up and red and itchy, kind of an annoying condition. So we think about inflammation as being kind of a bad thing, but it's absolutely necessary. If we have some kind of damage to our tissues, there has to be an inflammatory response before healing can begin. And hopefully I'll get you to understand how important it is. Through inflammation, that's how cells get to where they're supposed to go in order to fight invaders. Again, like I said, it has to happen in order for tissues to repair themselves. And it can be acute. That's when it's helpful. When inflammation becomes chronic, like this condition here, then it's not so good. And oftentimes in disease, it's the inflammation that really does the damage. Certainly been a lot of COVID patients where it's not necessarily the virus that's causing a lot of damage. It's just too much inflammation that the host puts out. So what's an appropriate level of inflammation, or why, when is it good? Okay, so that's what we're talking about. So when a patient has some kind of, say you got a cut in your arm, immediately the blood vessels in that area are going to constrict. They're going to get smaller just so the patient doesn't bleed out, right? But soon after that, because of that tissue damage, signals will be sent to make those blood vessels dilate and get a little bit bigger. Okay, so before there's some kind of, again, after that initial constriction, this is how the normal capillary bed would look. And then in the presence of some kind of inflammatory mediator, again, signals that might happen from some invader or some cut, then the capillaries will dilate. So we call this vasodilation. Okay, that those capillaries get bigger and they get leakier. What triggers this? What triggers the release of these inflammatory mediators? Remember back what I talked about last lecture, those complement fragments C3a and C5a, they can actually bind to some granulocyte. So maybe this is a neutrophil or an eosinophil or a basophil, and cause those granule containing cells to degranulate and release these inflammatory mediators. Your book may give you a few more examples of inflammatory mediators, but the one that most people have heard of is histamine. Okay, so that's an inflammatory mediator. And again, that's going to act on blood vessels to increase their permeability. Okay, and that that causes the cells that are in the blood vessels to be able to leave the bloodstream and get out into the tissues wherever the invader is present. So that ends up helps to resolve the infection. Okay. So your book's picture, this is very similar to the one that I showed already. Here's, it doesn't really show a great before or after, but right in the bloodstream, you've got your white blood cells, you got your red blood cells streaming through the bloodstream like really fast, remember? Okay, so if there's some kind of injury, cells like mast cells or neutrophils, we didn't really talk much about mast cells, but they have granules as well, release those inflammatory mediators. And now the junctures between the cells and the blood vessel wall will get larger. Okay, so by this process called extravasation, let me make sure that I write this correctly. Where did I write it? Okay, that's the process by which the cells move out of the blood, out of the capillaries into the tissues. Okay, so extravasation, all right? So here's a cell right here squeezing out between those cells that make up the blood vessel wall and moving out into the tissues. Okay, so this is going to happen much more frequently when those blood vessels are dilated and they've gotten bigger. Okay, and it's a complicated process because, like I said, these cells are moving through the blood vessels really quickly. And there's a number of different signals that have to be just met just right for those cells to slow down and stick to the ends of the blood or the sides of the blood vessel wall and then actually leave the bloodstream. There's a couple of YouTube videos that are in the text. One of them really gets into a lot of detail, and the other one is a little bit more accessible, I guess you could say, that talk about this process of extravasation. Another name for that is diapedesis. If you've heard that term before, I'll stick with the word that the book uses, and that's extravasation. So again, that's much more complicated than what we're going to talk about. Lots of signals so that the cells stick to the blood vessel wall and then can move out into the tissues. Okay. Now, if inflammation gets to be too much or too long acting, and you've got, you know, these big gaps in the blood vessel wall, what can happen? The red blood cells are not getting to where they need to go as quickly because the blood pressure goes way down. There's too much fluid leaving the blood vessels, and that can cause a condition known as shock. Okay, when the blood pressure gets too low, and that's potentially deadly. So again, you get too much inflammation all at once, and that's bad for bad state for the patient to be in. What are the effects that we usually associate with inflammation? There's 4 hallmarks. Sometimes we say 5, depending on the case, but we usually see pain. So due to pressure on the nerve endings. And you'll see all these effects if you just think about maybe somebody getting a vaccine or somebody getting an allergy shot. We usually experience all these 4 hallmarks of inflammation. Heat and erythema, that's another way to say redness. Those are both due to increased blood flow to the area. And edema, that's another name for swelling. Okay, so those blood vessels are getting leaky. We have lots of not only cells, but also fluid going into the tissues. And that's good on, again, kind of a short time frame because that helps get cells where they need to go. They can kind of hitch a ride on that fluid. That's probably quicker than moving by amoeboid action. It brings clotting factors if it's some kind of injury to clot up that injury. And if it's a pathogen maybe that's secreting some kind of extracellular enzyme or even some kind of toxin, then it's gonna, that fluid is going to help negate the effects of maybe any of those virulence factors. It does swelling typically lasts a long time. It takes a while for that extra fluid to kind of drain back into the lymph and get rid of it. Sometimes, depending on where this is happening, it can involve loss of function. Okay, that pain or the swelling might inhibit a patient's mobility. Here's an extreme example how inflammation can cause loss of function. That's somebody with the condition known as elephantiasis. That's caused by something called a filarial worm that hitches a ride on our public enemy number 1, mosquitoes. Okay, thankfully not around here, but in tropical regions. So again, that increased swelling would make it pretty difficult to move around. It can be prevented with like a yearly vaccine, but again, not everybody has access to that. All right, questions about inflammation? Okay, good, short term, usually not good, long term. Okay. Okay, we'll finish up our innate immunity. And remember, innate immunity, we think about immune function where we have the same response every time our body will encounter that pathogen. Fever, we could consider fever part of our innate immunity. It probably doesn't have a huge impact on fighting back a pathogen, but let's explore that. Anything that causes a fever, we would label a pyrogen. Okay, so that prefix pyro, that means fever inducing. And what kind of things, and actually a pyrogen is actually going to act on a place deep in our brain called the hypothalamus. That's our kind of thermostat for our body. Okay, so what kind of things can act as a pyrogen? It might be something that we encounter from our environment. So again, like a bacteria, that would be an exogenous pyrogen. Once that bacteria enters our body and enters our cell, then we would consider it endogenous. Sometimes parts of pathogens act as pyrogens, but very often it's our body's response to those pathogens, and it's our cytokines themselves that we release as in our immune system that act as pyrogens. Okay, so a little bit of fever, kind of short term, can be a good thing when those pyrogens are acting, without any damper on them. Then, uncontrolled, then that can be a very dangerous thing. What are the beneficial functions of fever? It can inhibit the growth of a pathogen. And probably most pathogens, not really very much. In lab, we usually do an experiment where we expose bacteria to warm or hot water baths for short periods of time. And it takes a pretty long exposure till of really high temperatures, like 50, 60, or even 70 degrees Celsius to inhibit pathogens, right? Our body temperature is 37. And if we have a fever, maybe it's 38 or 39, so it doesn't go up very much. Question, Jessica? Yeah, okay, we're going to talk about that. So should we take Tylenol? Okay, hold that thought. Okay, all right. One instance where it would inhibit a pathogen quite a bit, if we're talking about the pathogen that causes syphilis. Okay, that bacteria is really sensitive to any increased body temperature. So before we had antibiotics to treat syphilis, for a while, there was about 10 or 15 years that we were treating syphilis patients by giving them malaria, a really kind of mild form of malaria. So the guy that thought of that got a Nobel Prize. So kind of interesting. Apparently malaria is better than syphilis. Don't try that at home though. Okay, so maybe inhibit growth of the pathogen a little bit, maybe impede nutrient availability. When availability, yeah, just spelled it right. When we have a little bit of a fever, that's a signal to our cells to kind of sequester certain kind of nutrients that pathogens might need. So especially iron. Iron is often a limiting nutrient for bacteria. And so if our cells can sequester any available iron, then potentially that could inhibit the growth of some bacterial pathogens. Those two effects are probably minimal. We do know that fever does play a role in stimulating our immune system. Lots of aspects of our immune system. For instance, it increases phagocyte activity and we'll say the mobility of white blood cells. In general, the higher the temperature, the faster reactions happen. So some of our other aspects of immunity will kind of give them a little jump start when we have a fever. Having said that, though, the effect is fairly minimal for these things. And so from, as much searching and research into this as I've done, most experts say that it's probably fine to go ahead and treat a fever. So, when we have a fever, it certainly causes a lot of discomfort. Of course, kids are very cranky when they have a fever, and it's probably not worth the discomfort. And certainly when that fever gets to 101, 102, right, up into dangerous 103, we don't want it to get high. We don't want it to be uncontrolled, and we definitely want to bring it down. But even if you just have a little fever, go ahead and treat it. But it's just not, you're not going to get better any quickly by not taking something to reduce fever. So that's, there may be exceptions to that, but that's, again, mostly what my searches have come up with. Okay. I admit I was that mom that maybe withheld the Tylenol just so they would just lay there for a while. I might have done that a couple times when my kids were little. But yeah, no, you never gave Benadryl to put them out, but I thought about it. Yeah. Okay. So this is just a picture of where your hypothalamus is located. Don't worry about specific pyrogens or signals there. We didn't really talk about that whole process. So, okay, we're doing good. We're doing good. Let's move on. Okay. Let's at least introduce our adaptive immunity. I did, after I did this first slide, I kind of changed the order of things that I'm going to do. So don't pay too much attention to the order here. I'm thinking we will likely not get to vaccines. I'll make a decision on that on Wednesday, but there are some questions in your homework about 17.5, and I'll probably push those off, but I'm hoping to get to the rest of the chapter. Okay, so again, adaptive immunity is mostly dealing with B cells and T cells, and thinking about what they actually do and how they get activated, and the importance of this advertising system that I've alluded to, these MHC glycoproteins. Okay, so a little bit of an overview. How is adaptive, or what we consider adaptive immunity, different than innate immunity? This is our body's way to defend itself against distinct invaders. Okay, so I'm not talking about like 10 different PAMPs, pathogen-associated molecular patterns. I'm talking about millions of different things that you may never even get exposed to, but we have B cells and T cells with receptors that would probably recognize things like stingray venom. Okay, you're probably never going to be exposed to that, but we've got a few of them anyway, just in case. And then we would clone those cells, make many more of them. Okay, so the two most important attributes of adaptive immunity: specificity. Okay, so that's the ability of our, we'll just say, immune cells are going to respond to one particular shape. Okay, like I said, we have receptors to recognize almost anything. Okay, and the other important attribute is memory. Okay, so the next time an invader is encountered, we have a much quicker response. Because remember that. Okay, I had chickenpox when I was 8. I have probably been exposed to chickenpox many times since then, but I never even got sick because I have memory cells that recognize that right away and fought it off even before I got sick. So this chart, we're going to go into we're going to talk about this chart again later, but you have some initial exposure to a pathogen, and the first time after you're exposed, this is talking about how much antibody is produced, and hopefully you fight that off. But then the next time you're exposed to that, you have a lot more antibody produced. It's going to be the best kind of antibody, and it's going to get produced a whole lot sooner. Okay, so this is the basis of vaccines, that we expose ourselves to it once artificially, and then when we're really exposed to it, we have a much better response to it. All right, so let's talk a little bit more detail. We already talked about B cells and T cells in this lecture, so I don't feel like I have to maybe go I don't have to review very much. We just talked about that. But remember, we said that B cells differentiate into these plasma cells, and the plasma cells are what makes the antibodies. And by the way, B lymphocytes and T lymphocytes look pretty much the same. When you stain them, you wouldn't be able to tell the difference apart just from staining them. You would need some other, I don't know if they use antibodies or what they use to tell them apart, but they look the same under the microscope. The T cells, let me talk just briefly. Again, I think I mentioned this even again another time, but it's sort of basic foundational stuff, so we'll get it right. We have T helper cells, which we call TH. They orchestrate basically the entire adaptive immune response. Sometimes the other analogy that you hear is they're the generals in charge of the army. Okay, so the helper T cells are sending out cytokines and just kind of coordinating the whole adaptive immune response, the cytotoxic T cells and the B cells. Okay, so it's the cytotoxic T cells that are going to target and destroy host cells that are infected with some intracellular pathogen, like viruses. Some bacteria are intracellular pathogens as well, but viruses are always going to be intracellular pathogens. Okay, so, okay, all right, so we have to kind of assemble, talk about a bunch of things before we talk about exactly how the B cells and the T cells get activated to do those things. Okay, so what do they become activated against? Things called antigens. And I've tried not to use that term already before, but I probably have a few times. An antigen is just, let's see how to define this. Antigens are going to cause an immune response. Okay, so they're the parts of bacteria cells or viruses or fungi that elicit an immune response. Other things that are not a pathogen sometimes act as antigen. Some kind of food might act as an antigen, or maybe dust might act as an antigen. So, and then you have an inappropriate response to those, you have some kind of allergic response. Okay, so antigens can be things other than, protozoa or bacteria or viruses or whatever. The parts of antigens that are recognized by our immune system are called epitopes. Okay, so these are regions of antigen. We'll say that are recognized by antibodies, B cell receptors, and T cell receptors. Okay, so where here in this picture here, it labels like the whole bacteria cell, we could call that an antigen. But with that whole bacteria cell, there's many different epitopes that can be recognized by many different antibodies or different B cell receptors or T cell receptors. Okay, so really it's the epitopes that are part of the antibodies that are actually recognized. So what makes a good antigen? By that, I mean, what are the characteristics of a molecule that would make it really good at our immune system recognizing it? If you look at the shape or the complexity, that's one characteristic. Molecules that have a very distinct shape or are very complex are usually going to be noticed really easily. Okay, so the more complex it is, the more epitopes there are for our immune system to recognize. Also, the size is important as to whether our immune system recognizes it or not. Larger molecules are much more likely to be noticed than small molecules. And in fact, if the molecular weight is less than 500 Daltons, then it's usually not antigenic. Now, there are some exceptions to that. If a really small molecule complexes up with a larger molecule, then it can be detected. And we call those molecules haptens. Okay, so that's an antigen that's very small, that complexes with some carrier protein usually in order to get recognized. An example of that is poison ivy oil. So the active ingredient in poison ivy oil is called urushiol, and it's a really small molecule, so it shouldn't be antigenic. But if you've ever had, if you're allergic to poison ivy, then you know that it definitely can be. We definitely can have allergic reaction to that, and often do. But that urushiol complexes with some proteins in our skin to become antigenic. All right, so if we wash it off within a few hours of being exposed to poison ivy, then we don't get it. And by the way, you have to like scrub it with something, not just your hand. It's oil, so you have to kind of scrub it off with a cloth or something. But that's an example of an hapten. People that are allergic to penicillin is another molecule that's really small. But when we metabolize penicillin, and that small molecule again links up with the carrier protein for people that have an allergic reaction. Okay, so what kind of molecules are good antigens? Proteins are really good antigens because they're huge molecules and they're really complicated. Here's an example of, I just pulled an enzyme, beta-galactosidase. It's that enzyme that splits lactose. Just to appreciate the complexity of proteins, at least 200-300 amino acids. Those make really good antigens. So they stimulate, we'll say they stimulate the best immune response. Something that's a carbohydrate usually is less antigenic because carbohydrates are usually the same molecule like over and over and over and over again. So starch is just lots of glucose, 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 glucose. Okay, carbohydrates can be antigenic, but it's a little bit less common. And then lipids or nucleic acids usually are not antigenic unless they are combined with some other molecule. So like a lipid combined with a protein, a lipoprotein, that can be antigenic, or a lipid combined with a carbohydrate, a glycolipid, then that can be antigenic. Okay. And then we have some more terminology to do with antigens. We'll finish up with this. If antigens come from outside of our bodies, then we call them exogenous. Okay, so you know that's bacteria that you ingest with whatever, the hamburger that was undercooked. Okay, that's an exogenous antigen. And then endogenous antigens are inside of the cell. So they're produced within the cell by an intracellular pathogen. So something like a virus starts out as an exogenous antigen. We ingest it, it gets into our cells, its target cells, and now it becomes an endogenous antigen because it's inside our cell. Okay, so like I said, we have a way to advertise when our cells are infected that way that we'll get to. Autoantigens. These are particles of our host cells that can act as antigens. So normally autoantigens would not elicit an immune response, but there are some cases when things go wrong and our normal immune tolerance of ourself doesn't happen and we have an immune response. So something like if somebody has rheumatic fever, that's when you know, our body's joints are being targeted by antibodies. And that's not normal. It's when, you know, something goes wrong due to genetic or environmental factors. And lastly, there are super antigens. And we'll kind of touch base with this a little bit later when I can explain this a little bit more fully. But these are antigens that cause a way more than what's appropriate immune response. So many T cells are activated. We get like hyperinflammation, like 100 to 1,000 times more than what's appropriate. Okay, do a lot at you today, a lot more on Wednesday, so come back, but have a good day.